Okay. So on the female side, I have triple X. Okay, that's trisomy X syndrome. And most actually develop normally. And if you remember way back, we talked about something called a Barr body. And the Barr body is the inactivated X chromosome. And that kind of equals out the X genes. So trisomy X would actually have two Barr bodies, where other people would only have one. So that inactivation of that additional X chromosome actually allows for most to develop normally. And then we have Turner syndrome, which results in phenotypically short females. They usually have small breasts. They're often sterile, and they require hormone treatment for normal development. As far as some of the common alterations of chromosome number, this chart from your book shows frequency among live births. So as you can probably see here on the top is trisomy 21 or Down syndrome, and that occurs in about 1 in 1,000 live births, whereas the next most frequently seen are alterations in the sex chromosome number. One of the other common causes of chromosomal alterations is a deletion of genetic information. So in this case, we're looking at chromosome 5 on the right-hand side, and the area of C has been lost. That can happen due to maybe a virus or a radiation or some environmental factor. It can also occur spontaneously. But when one part of a chromosome is lost, the information on that particular piece of the chromosome is going to be compromised. So depends on what part is lost, what part is deleted, whether or not you'll see serious effects or maybe even lethal effects if the genes are vital to the organism. An example of this with chromosome 5 is something called Cri-du-Chat syndrome. That's where part of chromosome 5 is missing. In this case, we showed it as C. And that leads to abnormal development of the larynx, among other things. And the larynx is what we consider the voice box. The reason that we call this Cri-du-Chat means cry of the cat. And as a baby, the cry of a baby who has Cri-du-Chat, this loss of chromosome 5, sounds like a cat crying. It also leads to intellectual disabilities. Also, we can see translocations. And translocation is a little bit different. We are losing some of that information off one chromosome, but it's attached to another chromosome. So a translocation doesn't usually involve the actual loss of any genetic information. But in certain cases, it can, that translocation may move a gene or some genes to a different spot where that's more active, for example, so we transcribe it more, or it could fuse maybe with another gene. So that's actually what happens when we talk about chronic myelocytic leukemia caused by the Philadelphia chromosome. The Philadelphia chromosome arises from a translocation. We call it reciprocal because the information lost from chromosome 9 attaches to chromosome 22 and vice versa. So you're not actually losing any genetic material, but where it attaches, where one of them attaches, results in a different protein, and that protein is more active where it's located. So now it's very active, and we now have lost the ability to regulate growth and cell differentiation. We know that that leads to cancer, and so we refer to that fusion gene, that the two put together, as an oncogene. Remember, a proto-oncogene is a normal gene, but that once it is mutated or changed, then it can become an oncogene. So now it is a gene that makes a lot of a protein that makes cells continue to divide. In the case of leukemias, we know that leukemia is a cancer of the bone marrow. And as a cancer of the bone marrow, it's going to be located in the spongy bone at the end of long bones. And if you've done the tissues lab, you'll know what that means. And the bone marrow is where all our blood cells are made. So if we have a blood cell that gets out of control, like we do in chronic myelocytic or myelogenous leukemia, it starts growing and it overtakes the area that's supposed to be available for all our blood cells to form. So we see less red blood cells, which we know carry oxygen, and we see less platelets forming, and platelets are supposed to keep us from bleeding. They perform a function in clotting our blood. Also, remember all of our other white blood cells. We have a number of different types of white blood cells, and they all have important roles, and they're being swished out basically by this abnormal cell that keeps dividing. And the cell that keeps dividing, you'd kind of think, well, it's a white blood cell, it should still be fighting infection. But these are not normal white blood cells that go out and fight infection. They have lost that role. Remember, we talked about cell differentiation, and cell differentiation is where a cell has a particular role, and in cancer, that cell will start to lose the role that it was given, and that's what happens in the case of leukemia. All right, and now we're moving back a little bit to what we were doing at the beginning of this chapter when we were talking about homozygous and heterozygous and recessive and dominant. So put those back in your brain. And we're going to talk in this case about genetic information that leads to diseases. So when we talk about something being an autosomal recessive disorder, we're talking about it being passed on a regular chromosome, not a sex chromosome, so one of our 22 chromosomes. And when we say that it's recessive, we know that it can only be expressed when it is present in the homozygous state, so that we have 2 of those recessive genes. But in this case, we're talking about them being diseases. So when we're talking about these autosomal recessive disorders, males and females are equally affected. And affected children usually have normal parents. Why do they have normal parents? Because the disease is only expressed when there's 2 recessive genes. And if the parent has only 1 recessive gene, then their dominant gene that is normal is going to mask that recessive gene. So the parents will appear normal. Heterozygous individuals usually appear